Last session is CFAR Core's research interest and resources. So it's important that our cores better communicate and inform the CFAR users of the resources and services that we provide. You know, all of our cores are constantly trying to use information from the CFAR user survey and try to find better ways to help our CFAR members. So like this morning, Susanna mentioned the micro grants. That's a new strategy that we've put in place through the developmental core, and I've had some folks come up to me and talk to me about that. So this afternoon, we're going to hear from the other cores and learn about what resources that they have available to our CFAR users, and the first one is the Immunology Core. George.
Thank you, Herman. So the vision of the Duke CFAR Immunology Core is to enable Duke investigators to achieve an improved understanding of how to train the immune system to maintain health and prevent infection. The Immunology Core leadership brings together the expertise and experience of 7 scientists and their teams, Drs. Murdoch, Montefiore, Ferrari, Moody, Shen, Sarzati-Kelso, and myself from across 5 School of Medicine departments who provide innovative, state-of-the-art, and a standardized repertoire of immune assays, quality assurance consultation, and scientific training. And the organization of this dynamic core is led by Dr. Mudrick, a CFAR core facilitator. The achievements for the Duke HIV/AIDS research community are by the numbers for the Duke Immunology Core. We have over 100 investigators who utilize core immune assays and expertise. The top 9 core services include assays such as binding assays, antibody Fc effector function assays, neutralization assays, PK, as well as consultation related to quality assurance and tools for quality assurance within the laboratories. The Immunology Core has fostered collaborations for high-impact science and HIV incidence, next-generation therapeutic and cure strategies, HIV-associated comorbidities, and maternal immunization strategies. Together with collaborators, they have published over 60 publications and contributed data and expertise to numerous CFAR pilot and NIH grants. And notably, the Core has mentored and trained over 31 Duke early career investigators who have now gone on to design studies, receive funding awards, and progress in their careers. So we wanted to highlight 2 projects utilizing our time to talk about the Immunology Core. The first is from a partnership of the Duke CFAR in 2018 that partnered with the Fred Hutch to co-host the Mucosal Immunology Program. The Mucosal Immunology Program was initiated in 2009 by the Fred Hutch to provide small grants to investigators working in the mucosal immunology field and to develop methods and standardize them for use across the field. Notably, Primavada Acharya applied for this funding when it was hosted here through the Duke CFAR and was awarded a MIG grant to study the effect of mucosal fluids on HIV envelope structure, as well as classify endogenous antibodies within mucosal fluids, and worked with a team of investigators I have listed here. This project has yielded important information to date through the development of an automated 2D class average classification system using deep learning methods to increase assignment consistency and enable a robust quantitative description of the results. So we're really pleased about this outcome. And for further information, you should see the poster from Shana McDowell and collaborators who had a poster here at the Duke CFAR. And this work was enabled by the work with the CFAR Immunology Core scientists and the biostatistics and computational cores that you'll hear from next. And next, I'd like to invite Tinashe Nayan-Hedi, who will give a short talk about his exciting results from work that's a collaboration between the Duke CFAR Immunology Core and the clinical cores. Tinashe.
Thank you. So one of the many things we've been doing with the HIV virus controller patient cohort is trying to look at the presence of broadly neutralizing antibodies, or BNAbs, in the scenic patient group. So HIV virus controllers, these are HIV patients that have the natural ability to control infection without therapy. And we think that this patient group actually provides us with a nice model that actually allows us to be able to actually study the different factors that are associated with controlling HIV-1 infection. And hopefully down the line, this will actually help better inform HIV-1 vaccine design efforts going on. So with the help of the CFAR Immunology, CFAR Clinical Core, we actually have 57 patients enrolled in this patient cohort. So for the virus control patient cohort, we have the enrollment criteria Firstly, the patient has to have a viral load that's less than 5,000 copies per mil. They also have to have a CD4 T cell count that's more than 400 cells per microliter. And they also have to not been exposed to any sort of treatment. And looking at the patient distribution, we have 28 males in the group, 28 females, 1 transgender female. We also have a wide range in age, ranging from 14 to 75 years old in this patient group. So as I mentioned before, we're interested in determining the presence and the potential protective role of bNAbs in this patient cohort, which is an interesting topic because it's always been widely accepted that patients who have bNAb activity, they tend to have very high viral loads. But that seems not to be the case anymore, since we have a few recent studies that are actually showing that you can actually detect and even isolate bNAbs in patients with very low HIV viral load. So for us, our group, we're more interested in trying to define the virus controller bNAb specificities and also the frequency of bNAb development in this patient group. So far, we have seen that 6 of the 31 patients we have tested actually have bNAb activity. So that represents 19% of patients in this patient cohort. And the criteria we used actually determine whether a patient has bNAb activity was firstly looking at neutralization magnitude, which was determined or which was measured as a geometric mean ID 50. And we also looked at the neutralization breadth coverage. So for these 6 patients who have bNAb activity, which are the patients above the black dotted line, they have a geometric mean ID 50 of more than 100, and they all neutralized 90% of all the viruses that they were tested against. So now the next question was then trying to define the epitopes that are actually being targeted by the polyclonal antibody responses in the 6 patients that have bNAb activity. So one approach that we used to determine the epitope specificity was actually looking at the neutralization signature of a patient plasma sample and then using the least squares fitting model, which is a regression analysis, to then try and see or compare how similar that neutralization signature compares to the neutralization signature of 10 known bNAbs that actually bind to 10 different vulnerable sites on the HIV-1 envelope. So similarity in the neutralization signatures between a given patient plasma sample and one of the 10 bNAbs that we were comparing them against to was then given as a number that was a ranking from 0 to 1 with any number above 0.25 representing a positive signal. So using that criteria, we saw that 5 of the 6 patients we have had a PGT128-like activity. So this is binding to the V3 region. And then 4 of the 6 patients we looked at had 10E8-like bNAb activity. So this was binding to the AMPA region. And then 2 of 6 patients had VRC01-like activity, which is binding to the CD4 binding site. So the PG128-like activity, which was the more prevalent activity we detected in this patient group, wasn't really that interesting for us because that activity has actually been seen in multiple VC patient cohorts where they actually screen those patients for bNAb activity. However, of interest for us were the 2 patients where we saw VRC01-like activity that's binding to the CD4 binding site, because with this assay, you normally don't pick out a VRC01-like activity when you use this assay in different patient samples you have. So to actually give you guys a much more better context of how this is an interesting finding, this VRC01-like binding activity, we also ran the donor sample where they actually isolated VRC01 bNAb using this assay. And when we ran that assay, we actually saw that for that donor sample where they got VRC01-like, the score they got was 0.4. And when you look at the 2 patients we have, when you run this assay, the scores we're getting for one of the patients with C39 is 0.4, which is comparable to the score you get when you actually run this assay with the donor. We actually isolate VRC01. And then the other patient with C38 actually has a score of 0.5, which is even higher than the score you're getting from the VRC01 donor. So I think this was very interesting that you actually have this activity being predominant in these 2 patients, and this is actually a rare finding in other patient cohorts where they've used this assay. So despite this interesting finding, one of the major limitations with this computational approach is that it only looks at the binding to already predefined epitopes. So one of the things we're trying to do is we're trying to see that the bNAbs are actually getting from the virus control patient cohort, are they binding to other undefined epitopes on the envelope, which might mean that maybe they have novel binding that actually leads to protection. So with this assay, since they're only comparing binding to already predefined epitopes, you won't be able to actually define or identify novel binding epitopes. So to overcome this limitation, the other approach we're using is utilizing high-resolution imaging. So in this instance, we're using negative stain electron microscopy and also cryo-electron microscopy. So with this approach, what we do is we isolate the plasma samples from the 6 patients where we have BNAB activity, generate our IgG FAB fragments from the plasma samples, and then use the FAB fragments to form immune complexes with HIV envelope trimers of interest. And once we have those immune complexes, we then use the high-resolution imaging technique to actually then see where the FAB fragments are actually binding to on the HIV-1 envelope. So with this approach, we hope that it actually help us uncover any previously undefined epitopes that might be unique to the virus control patient cohort, which we think might also play a role in mediating viral control in this patient group. So to summarize what I have, or what we've found so far, is we know that we have a subset of HIV-1 virus control patients that do possess BNAB activity, and we also know that these BNABs that we're seeing in this patient group, they're actually targeting some of the known vulnerable epitopes on the HIV-1 envelope, suggesting that the BNAB development in this patient group can actually occur in the absence of high viral load, which is something that's also been confirmed in other patient cohorts. Moving on, the plans moving forward is to try to actually then identify the vulnerable epitopes that are being targeted by these VC antibodies using the high-resolution imaging techniques we're trying to employ. And once we have those epitopes identified, then the next thing is then to try and see whether, by looking at those epitopes and looking at the sequences in those regions, do you see any mutations in those sequences that then gives you an indication of whether you have immune pressure being exerted in those regions. And that could act as a proxy to actually get a better sense of whether the autologous humoral response or antibody response in those patients actually does something to the autologous virus in that same patient. So that's what we have so far, and hopefully moving on forward with all the people we're collaborating with, we should be able to have more exciting data to present down the line. And so I'd like to thank all the people who have been involved in this group, especially the CFAR Clinical Core, for having us, for granting us access to the patients that come to the hospital, and then the other Immunology Core facilities for the techniques and expertise that they offer in doing these basic science studies in the lab, and the different finding sources that we've been using to actually support this work. So take any questions if you guys have any.
Tanashi, that's fantastic. Of your 6 patients, the subset, would any of them fall into a classification of elite controller where you've got almost non-detectable viral loads, or do they all have, are they hovering around 5,000 or a little bit below?
So they're hovering around, so the average is around 2,000 copies per mil. So the other thing I didn't show you that we did was, within the 31 patients we do have elite controllers, so less than 50 copies per mil, and we have virus controllers. So we have 21 virus controllers, 10 elite controllers. So when we did the neutralization assay, we actually tried to stratify and see whether there's any differences in the breadth and magnitude of the neutralizing activity. So I actually see lower neutralizing magnitude and breadth within the elite controllers and slightly higher in the virus controllers. So it seems like you do have to have a certain level of viral load to actually induce or stimulate BNAB development. If it's very low, most likely you won't get anything. And there's 6 patients, they're all virus controllers. They're all about 50 copies per mil. So it seems like viral load still plays a role, but it doesn't have to be that high as what you find in chronic progressors. Yeah.
So in this cohort of patients, I noticed you identified these 6 that have broadly neutralizing antibodies.
Yeah.
What type of immune responses are in the other subjects that don't have broadly neutralizing antibodies that contribute to controlling the virus?
So just within the virus control patient cohort in general, we know that there's heterogeneity when it comes to immune responses. So some have strong CD8 T cell responses, some have neutralizing activity, some have strong ADCP activity. We're also looking at mucin binding, which is, we think it's another novel Fc effector function that could play a role in all this. But we know that there's different immune responses that they have, and in other cases, it could be the virus itself. So it could be a defective virus that actually infected the patient that makes it harder for the virus to replicate and hence have the virus controller status. So it's a different mix of different things, immune responses, genetics, and the virus itself too playing a role in mediating control. Yeah.
So the next, thank you.